Hello, and welcome to this Microbiology Bio 203 video lecture on Chapter 7 from your OpenSTAX book, Microbial Biochemistry. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Learning outcomes for Chapter 7 include being able to define the building blocks of life, or biomolecules, such as carbohydrates, lipids, proteins, and nucleic acids. As we explore biomolecules, we'll begin by taking a closer look at carbon. In order to be considered a biomolecule, or for us to study organic chemistry, we have to understand the basic properties of carbon. Carbon is at the heart or the backbone of every biomolecule or every molecule in organic chemistry. The four most common elements in living matter are carbon, nitrogen, oxygen, and hydrogen. Of the four, again, the most important is carbon. A carbon atom can form four covalent bonds, making it a unique atom in the chemical world. This allows it to build large and diverse organic compounds. Molecules that possess only carbon and hydrogen are referred to as hydrocarbons. They tend to be energy-rich and are nonpolar. Most biomolecules also contain a variety of functional groups, which adds to their unique characteristics. The carbon skeleton. The carbon skeleton adds diversity to biomolecules. Carbon can form single and double bonds. It can form long chains with simple and double bonds, branched chains, and even rings, all as illustrated on this slide. Here are some examples of organic molecules that we'll deal with as we go through this presentation. You can see the architectural differences between the molecules as mentioned on the previous slides. Some are long chains, some have rings, and in more complicated molecules, you'll find combinations of the two. Carbon can form single bonds, which leads to a tetrahedral shape. When it forms double bonds, we get a planar shape. Organic molecules or biomolecules may also have isomers of one another. In the case of an isomer, we have the atomic makeup of the biomolecule being identical from one molecule to the next, but having the atoms organized into slightly different structures. When we have different bonding sequences, we refer to biomolecules that again share the same atomic makeup as structural isomers. If they share the same atomic makeup but have different spatial arrangements, then we refer to them as stereoisomers. The functional groups of biomolecules again provide distinctive properties to those organic molecules, and they depend on the carbon skeleton and on the chemical groups attached to it. A functional group is a group of atoms bonded to a carbon of an organic compound, giving a property such as polarity or acidity to the biomolecule. Some examples are listed here, such as the COOH group or carboxyl group, which imparts an acidic behavior to the biomolecule, or the OH alcohol group. This table will provide you with a list of the simple functional groups used in organic chemistry, their structure, and some of their basic properties. Let's explore them further. Here we have the hydroxyl group or OH group. It is common in organic alcohols. Some examples include ethanol, which is listed here in its chemical formula. It's also present in sugars and can contain one or more OH or hydroxyl group. The carbonyl group is a carbon double bonded to an oxygen, and it's illustrated on this slide in several forms. There is the aldehyde, which can be found at the end of a chain, a ketone, which is not at the end of the chain. Carbonyl groups can be found in sugars such as aldose and ketoses. The carboxyl group is next. As mentioned previously, it imparts an acidic nature to the biomolecule which possesses it. So it's not unusual to refer to a biomolecule that has a carboxyl group as an organic acid. Some examples include vinegar and acetic acid. Amino groups. The NH2 is the amino group, which gives the molecule a basic or alkaline characteristic, and it is the primary building block in an amino acid. The sulfohydryl group, or SH group, is also common in amino acids, as seen here in cysteine. The phosphate group, or PO4 group, tends to carry a negative charge and is commonly found in nucleic acids, such as ATP, and phospholipids. The methyl group, or CH3, is found added to a variety of different molecules. Adding the group may change the character of the molecule. An example here is adding methyl groups to DNA, which makes DNA less accessible and silences it. Next, we'll look at the difference between monomers and polymers. In lay terminology, a monomer is analogous to a brick, while a polymer is analogous to the entire brick wall. Most biomolecules are built of smaller subunits that are referred to as monomers. Polymers contain many monomers. Some examples include monosaccharides, which are monomers for polysaccharides, and amino acids, which are monomers for proteins, nucleotides, which are monomers for nucleic acids. Dehydration synthesis reactions are common in building polymers from monomers, while hydrolysis reactions break them down. This is an illustration of hydrolysis and dehydration. Adding water breaks polymers into monomers, while the removal of water makes polymers out of monomers. The molecules of life are next. Here we will explore carbohydrates, lipids, proteins, and nucleic acids. We'll begin with carbohydrates. The three primary atoms responsible for the base structure of a carbohydrate are carbon, hydrogen, and oxygen. They tend to be found in what is referred to as the CH2O to the N empirical formula. The major function of a carbohydrate is to supply energy, or to be a source of cellular food. Monosaccharides, disaccharides, and polysaccharides are all examples of carbohydrates. Monosaccharides are the most simple, disaccharides are a combination of two monosaccharides, and polysaccharides are the most complex. Here we have monosaccharides. Monosaccharides tend to be made of 5 to 6 carbon structures. The 6 carbon structures are referred to as hexoses and can be found in glucose, dextrose, and fructose. The 5 carbon pentoses are common in ribose and deoxyribose. Monosaccharides present carbonyl or C double bonded O groups and hydroxyl groups and can be ketoses or aldoses. The disaccharides are made by binding or bonding two monosaccharides together. In the case of table sugar or sucrose, we bond glucose to fructose. Lactose is made by bonding glucose and galactose. Maltose is made by connecting two glucose molecules. Polysaccharides are large polymers of sugar. Simply put, you need 3 or more monosaccharides bound together to make a polysaccharide. They can be used for structure or storage. They can be straight, helical, or branched. In their storage form, they can be found as starch in plants or glycogen in animals. In their structural form, They can be found as things like cellulose in plants and chitin in arthropods. Here are some pictures to compare some different polysaccharides. Modified glucose molecules are next. In bacteria, glucose can be modified in some unique ways. The bacterial cell wall contains N-acetylglucosamine, or NAG, and N-acetylmuramic acid, NAM, which are part of the peptidoglycan cell wall. Polymers of NAG can also form chitin. Next, we have lipids, or fats. Lipids, or fats, contain the same three base atoms as do carbohydrates: carbon, hydrogen, and oxygen. However, the empirical formula here is different. Lipids are all hydrophobic, which means that they do not interact well with water. The reason being is that they lack an overall charge. Some examples of lipids include the neutral fats or triglycerides, phospholipids, waxes, steroids. There are no monomers in lipids. The first lipid to consider is a triglyceride, which is made of glycerol and 3 fatty acids. If one were to attempt to describe a monomer of a lipid, this would be your first go-to. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. Attached to the glycerol are fatty acid chains. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. The length of these chains is highly variable. Here are some examples of triglycerides being formed through dehydration. The dehydration reaction occurs between the alcohol and the fatty acids, where water is removed. Fatty acids can exhibit long carbon skeletons plus a carboxyl group. They are, again, hydrophobic or nonpolar. If it contains only single bonds, the fatty acid is referred to as saturated and tends to form solids at room temperature. If it contains double bonds, then it is described as being unsaturated and will often be liquid at room temperature. Phospholipids. Phospholipids are one of the most important lipids that we study in any science class. Phospholipids are modified triglycerides with only 2 fatty acid groups and a phosphorus group attached. Their structure allows them to interact with both water and fats. They are directly responsible for the formation of the phospholipid bilayer that makes cellular life and all life on Earth possible. This is an illustration of how the phospholipid bilayer forms. Isoprenoids. Isoprenoids are modifications of isoprene, which play multiple roles. They can be used as pigments, fragrances, and pharmaceuticals. Steroids. Steroids are flat molecules with 4 interlocking hydrocarbon rings. The main example that you should familiarize yourself with is cholesterol, as it is directly responsible for adding stability to a cell membrane. Next, we have the proteins. Proteins contain carbon, hydrogen, nitrogen, oxygen, and some sulfur and phosphorus. Their monomers are amino acids. There are 20 different amino acids that are used in some combination to produce every protein known on Earth. Proteins can perform a variety of functions. They can act as a catalyst or enzyme. They can be transport molecules, structural components. They can be used in communication. They can be receptors for hormones. They can also be used for defense. Antibodies and clotting factors are also proteins. The sequence of amino acids is the information coded in the genes and determines the behavior and structural form of the protein. Here we have amino acids. there are, again, 20 amino acids that are largely responsible for every protein that we know on Earth. These amino acids contain an amino group and a carboxyl group. The amino group, or NH2 group, is on one end of the amino acid, while the carboxyl group, or COOH, is on the other. In the center, you will have a carbon atom attached to a functional group or side chain labeled R in this picture. Amino acids can be polar, they can be nonpolar, and they can be charged depending upon the behavior and architecture of this side group. The peptide bond is what holds amino acids together. The NH2 or amine group of one amino acid can bind to the carboxyl or COOH group of another and form a peptide bond through dehydration synthesis. Less than 50 amino acids equals peptides. Many amino acids bound by peptide bonds is considered a polypeptide or protein. Once proteins form their base structure or amino acid sequence, they will often fold and take on three-dimensional shapes. In their base or simple amino acid sequence architecture, we refer to them as being in their primary protein structure. As they start to fold and take on three-dimensional shapes, they will move through secondary, tertiary, and ultimately quaternary modifications. Secondary modifications to structure include forming alpha helices and beta-pleated sheets, while tertiary structures start to develop some three-dimensionality as the alpha helices and beta sheets become superimposed on each other. Finally, in quaternary structure, polypeptide chains are linked together in a specific manner, oftentimes with prosthetic groups that add unique behaviors to the protein. In protein architecture, we will sometimes find non-protein groups that are either lipids or carbohydrates, which make lipoproteins and glycoproteins. Altogether, protein structure determines the behavior and function of the protein. Here are some examples of protein going through some architectural changes. This is primary structure. Here we see secondary structure, tertiary, and quaternary structure. Here's a single slide to summarize the movement through all stages of protein development. Native structure. Proteins require its native structure for correct functioning. That native structure is the shape that it needs in order to do its job. As with any other quote-unquote living thing, we all have conditions under which we prefer to work, such as an appropriate temperature or pH. Proteins are no exception to this rule. If a protein is superheated, it can denature or unfold, at which point it loses its native architecture and will no longer function. Cooling a protein down does not destroy its native function, but can also adversely affect its ability to do its job. Denaturization can be reversible or irreversible, dependent upon the stage to which it is carried. Using biochemistry to ID organisms. Here we will look at biochemical tests that can detect unique molecules. Commercial systems may identify multiple reactions in an automated way in a form of an experiment called a biolog. Mass spectrometry can also be used to ID organisms. Mass spectrometry compares the chemicals released from the microbe after UV radiation to a database. Matrix-assisted laser desorption ionization time-of-flight mass spectrometry is an example that is often used in microbial ID. Lipids can also be used to ID organisms. Many organisms have unique lipid profiles. Using fatty acid methyl ester or FAME analysis, organisms can be ID'd by their lipid profile. Proteomic analysis analyzes or looks at proteins produced under specific conditions for the same purposes as the previous mentioned experiments to ID and classify an organism. Proteins will be separated, digested, and analyzed, and then compared to a database. This brings us to the end of Chapter 7, Microbial Biochemistry.